Hello everyone, welcome to today's event. I'm Brian Demansky, Strategic Collaboration Scientist at Shimadzu Scientific Instruments. I'll serve as your host for our discussion about the applications of centrifugal field flow fractionation to nanoparticle drug delivery systems. Before we start, I want to share a couple of notes for our viewers. The webinar console has a variety of items to help enhance your experience and interaction with us. In the screen, you'll see the following items. The slides will appear in the center of the console. Directly under the slides, you will see the relevant content list with clickable links relevant to the material being presented today. On the top left of the console is the question widget. Please submit your questions during the presentation through this widget, and we will answer them during the Q&A session. Just below the question box are survey questions that you may fill out anytime during or after the presentation. If you would like to be contacted by a Shimadzu representative directly, use the Request Demo section on the right to start that process. The icons at the bottom of the screen can be used to restore any of the widgets in case they are minimized or hidden. Our speaker today will be Nivesh Mittal, who currently serves as the Liquid Chromatography Product Manager at Shimadzu Scientific Instruments. Without further ado, we welcome Nivesh to introduce his presentation and his background.
Thank you so much for the introduction, Brian. Hello, everyone. My name is Nivesh Mittal, and like Brian said, I am the current LC Product Manager for Shimadzu Scientific Instruments. I also have an extensive background in the development and manufacturing of nanoparticle drug delivery systems in my past roles as a formulation scientist, and I hope to use some of that today to increase your knowledge about centrifugal field flow fractionation and how that can be used to separate nanoparticles and overcome the challenges of existing technologies. Before we start, quickly, here is the agenda. We will start with a short background on the nanoparticle field, where field flow fractionation is used the most, and what are the challenges in nanoparticle analysis. I will then talk about field flow fractionation technology and the Shimadzu CF3 C8030 system. We then compare CF3 with other field flow fractionation technologies, such as the AF4 or asymmetric flow field flow fractionation. And finally, I will show you some of the applications using CF3 as well. So let's get started with a brief background on nanoparticles and their general applications. So what are nanoparticles? They are ultrafine particles, typically between 1 and 100 nanometers, and sometimes up to a micron even. Just to give you a perspective of how small they really are, a single strand of DNA is about 2 nanometers, an antibody molecule is about 10 nanometers, and a virus particle is about 100 nanometers. A nanoparticle is to a soccer ball what a soccer ball is to the Earth. Hopefully that gave you an idea of how small nanoparticles really are. So what's so special about nanoparticles that we are talking about them so much today? Well, the first thing that is special is their surface area. The surface area of nanoparticles can be 1 million or even 10 billion times higher than its summated volume structure. This can make them highly reactive and render them with other altered physical properties, such as special optical, thermal, mechanical, and magnetic properties. Nanoparticles have become very, very popular for their application in medicine as well. There is something called the EPR effect, enhanced permeation and retention effect, and nanoparticles are basically able to enter diseased tissues in the body and accumulate there in high concentrations because of poor lymphatic drainage. This property can be used for targeted delivery. This also causes reduced adverse effects from medicines. They can be used to formulate long-acting medications as well. There are several long-acting anti-cancer or antifungal medications on the market today that are nanoparticles, that have nanoparticles in their formulations, basically. Nanoparticles are also used in material science and the electronics industry, such as the manufacturing of computer chips or semiconductors, flexible displays, creating stronger but lighter materials, such as components that go into carbon fiber bikes and self-cleaning surfaces. There are 3 primary types of particles used in the biopharma industry: liposomes, polymeric nanoparticles, including solid lipid nanoparticles and metallic nanoparticles. Liposomes are made of a phospholipid bilayer and have an aqueous core. The drug molecule can be in the aqueous core or in the hydrophobic lipid bilayer. Examples, of course, everyone knows COVID vaccines and Doxil, which is an anti-cancer, one of the first nanoparticle drugs to be approved for an anti-cancer as an anti-cancer molecule. Polymeric nanoparticles typically have a core shell type structure. The core is polymeric, and most often the drug loaded is in the core. The outer shell can be a lipid or a polymeric shell, and the example is Refixia, which is an FDA-approved anticoagulation factor drug product which is used for bleeding disorders. Metallic nanoparticles, of course, have a metallic core and several stabilizing layers around it. Layers of drug can also be loaded for drug delivery purposes. However, their most common application is in GI imaging for MRIs. The most common techniques that are used in the analysis of nanoparticles are microscopy techniques that give information on the size, shape, and the surface morphology of the nanoparticles. Light scattering techniques that can give information on the size and the charge of the nanoparticles. SEC or size exclusion chromatography, as we already know, separate particles based on their size alone. And finally, field flow fractionation, which can separate particles based on their size and or density, depending on which type of field flow fractionation we are using. So what are the challenges that scientists encounter when using these analytical techniques for particle separation and the other types of different analyses? Let us look at SEC versus hydrodynamic chromatography first, or HDC. SEC uses porous particles, which have the benefit of being able to analyze a broader range of sizes, from sub-nanometer up to 200 nanometers even. HDC, on the other hand, uses non-porous particles, and so the range of effective resolution is in higher size nanoparticles, up to micron-sized particles, depending on the column used. SEC suffers from non-specific interactions with the column, and no morphological information, such as drug-loaded or empty nanoparticles, can be obtained. It's purely based on the size alone. Hydrodynamic chromatography, on the other hand, has drawbacks such as lower resolution due to the larger pore sizes between the particles. The potential for column clogging is also higher with HDC. Using electron microscopy, the particles can be most effectively analyzed for their sizes. It is the most accurate for the size. However, if the sample has particles of various sizes, then the smaller particles tend to get obscured by the larger ones, and the results are inaccurate. Sample preparation for electron microscopy is also not easy, taking multiple days for some of those techniques. Dynamic light scattering, or DLS, is also a user-friendly method to analyze particle size and distribution. Unfortunately, it gives a particle size distribution and a mean size only. It is not able to differentiate between closely similar sizes within a sample. So if the sample has a polydisperse or a variety of different sizes, the results will be inaccurate. Field flow fractionation, or FFF as it is commonly known, it is a separation technique that can address the shortcomings of your standard chromatography or microscopy methods, ensuring good separation performance as well as being able to assess a wide range of particle sizes. In FFF, separation is achieved by applying a field or a cross flow perpendicular to the direction of carrier flow as the sample is pumped through a long channel. Other techniques either lack separation performance, such as DLS or the microscopy techniques, or they lack a broad size range, such as the chromatography techniques. Here we can see a summary of the different particle size analysis techniques and the ranges that they are most suitable for. Today, however, we will be focusing on the principles of field flow fractionation alone and how Shimadzu has developed a solution for this field. Let us look at the science of field flow fractionation in general and then the Shimadzu's CF3 system. Field flow fractionation is basically a family of elution-based separation techniques. It uses a force field instead of a column to push the sample particles in one direction, while diffusion forces push the particles in the opposite direction. Maintaining a higher force field compared to the diffusion forces the particles to separate and elute. There are several types of field flow fractionation techniques, the most common one being AF4 or asymmetric flow field flow fractionation, primarily because of its versatility with different nanoparticle types and samples. However, it is unable to distinguish between drug-loaded and placebo nanoparticles, and that is where CF3 or the Centrifugal Field Flow Fractionation unit has an advantage. I will discuss more comparing the two techniques in a few minutes. Thermal, electrical, and even gravitational FFF exist, but they are not so commonly used compared to the CF3 and AF4. And now, introducing the Shimadzu CF3 C8030 Centrifugal Field Flow Fractionation System. It is capable of analyzing a wide range of particle sizes, and it offers the best resolution using centrifugal forces of up to 15,900 Gs or 12,000 RPM. The Shimadzu CF3 is the first to achieve lipid nanoparticle separation using CF3. It can be used with various detectors and collect fractions and further analyze them on an electron microscope. The range for the particle analysis is from about 10 nanometers to 20 microns. Let's walk through the standard separation principles of our CF3. And how exactly do these specifications help with overcoming the limitations of conventional particle separation technologies? The high centrifugal speed assists with achieving higher resolution between particles of closely similar sizes. The size range is wider than other technologies, offering from low nanometer to multi-micron separation capability. There is no chromatographic media, as in SEC or HTC, for non-specific interactions to take place. Drug-loaded and placebo or empty liposomes or nanoparticles can easily be distinguished based on the difference in their masses. And finally, even if the sample has nanoparticle populations of a variety of sizes, separation will consistently and effectively be achieved, unlike DLS. Think of it as a regular LC system, but instead of a column for separation of molecules, we are using the CF3 for the separation of particles. We use the same pumps, the same autosampler, and even the same detectors can be used. Just instead of the column and the column oven, we have the CF3 system now. The separation takes place on the CF3 and the detection is done using a UV absorbance, fluorescence, RID, or a MALS multi-angle light scattering detector. The separation principle is as follows, and I briefly did speak about it. The separation channel rotates around a central axis at a high speed. The nanoparticles all hit the wall initially. As the centrifugal speed slows down, the smaller particles leave the wall first and elute first, and the largest particles elute last. The most common detector that is used for particles coming off a CF3 would be the MALS multi-angle light scattering detector. But in the absence of a MALS, other detectors can also be used, such as a UV absorbance, fluorescence, or an RID. They would provide lesser information though. Samples coming off the CF3 can be collected using a fraction collector, and the fractions can be used for microscopy techniques like electron microscopy or other nanoparticle analyses such as zeta potential or surface morphology, say for example through AFM. And now, let us compare CF3 with the AF4 or the asymmetric flow field flow fractionation technique. So AF4 works using a membrane. The carrier flow is parallel to the membrane and a small pressure is generated inside the separation chamber. By controlling this pressure, the carrier flow rate, and the porosity of the membrane, a membrane filtration of the particles is achieved, with the smaller particles eluting first, followed by the larger particles. CF3, on the other hand, works completely differently using a centrifugal field, as mentioned. As we all know by now, the separation on a CF3 is based on the differences between the masses of the nanoparticles. AF4, on the other hand, separates purely based on the size of the nanoparticles. The separation range for CF3 starts at about 10 nanometers and goes up several microns. For AF4, it starts a little lower than the CF3, and the upper limit is also a little lower than the CF3. The analysis time for the CF3 is 30 minutes to several hours, depending on the resolution that is desired and the nature of the sample. AF4 analysis is up to 100 minutes typically. Benefits of the CF3 are primarily on the basis of separation, which is the mass of the nanoparticle. Analytical conditions can also be easily developed. AF4, on the other hand, has challenges with the membranes being prone to fouling and non-specific interactions. Analytical conditions are also tough to develop, with several parameters needing optimization, such as membrane porosity and type, carrier flow and pressure, among others. The primary advantage of the AF4 is the versatility of the sample type that can be analyzed compared to the CF3. One of the most common advantages of using CF3 over AF4 is the possibility to differentiate and separate drug-loaded and empty nanoparticles. As you can see in the results here on the left, the drug-loaded nanoparticles and placebo nanoparticles have the same size, but they can still be separated by the CF3 system based on their differing densities. The first peak is for the lighter but the same size of the empty liposomes, followed by a distinct separate peak for the drug-loaded and heavier liposomes. In contrast, AF4 is completely unable to achieve this kind of a separation. I just want to show some data to explain how the higher centrifugal speeds result in a higher resolution. The study here uses silica nanoparticles ranging from 30 to 70 nanometers in size. The figure on the left are results obtained running the Shimadzu instrument at a lower speed, that is 4,900 RPM, and the figure on the right is using the same sample but acquired using the highest RPM on the Shimadzu system, that is 12,900 RPM. On the left side, we can clearly see the suboptimal resolution between the sizes and the smallest size almost coming out in the void volume. The higher speed results on the right-hand side show baseline separation as well as the smallest size eluting away from the void. This demonstrates that the higher centrifugal speeds of the Shimadzu system are necessary to achieve higher resolution and higher sensitivity when separating and analyzing nanoparticles. Just a few more applications now. The CF3 CAD30 system can also be used to simply prep the samples for analysis by microscopy techniques such as SEM or TEM. On the top left, you can see the sample of silica nanoparticles has populations of multiple sizes. Using the CF3 CAD30 technique, they were effectively separated and collected using a fraction collector. Next, they were analyzed using SEM once again, showing clean sample populations of a single size only. Particle separation before analysis helps in getting more accurate information about particle morphology, which in turn helps to make better informed scientific decisions. Another very interesting study evaluates drug release from nanoparticles using the CF3 CAD30 system. It also studies interbatch and reconstitution variability. These are all critical quality attributes or CQAs for the analysis of nanoparticle drug products. Summarizing today's talk, we discussed several things. The Shimadzu CF3 system has the highest rotational speed on the market that helps with analyzing smaller particles and achieving a high resolution between them as well. It overcomes several limitations of the conventional particle analysis technologies such as SEC, electron microscopy, and DLS. It enables connection of the separated particles and downstream analysis using other techniques. And most importantly, it is also capable to separate drug-loaded nanoparticles from empty nanoparticles. These features make this the ideal addition for your lab if you're working on nanomaterials or any other novel biologics. Thank you so much for joining me today to learn about this technology and the Shimadzu system. And I can now take a few questions. Thank you.
Let's thank Nivesh for such an engaging presentation. As we prepare for the Q&A session, I encourage you to submit questions for the discussion through the question widget in the upper left of the console. If you want to learn more about centrifugal field flow fractionation applications, please check out the related content list. And if you've not had the chance, please fill out the survey questions on the left-hand side to provide feedback and request additional information, including the ability for you to sign up for our Shimadzu e-newsletter. That once monthly blast includes information regarding upcoming webinars and events that encompass a wide variety of application areas. At this time, we will begin our Q&A session. So again, thank you for your audience participation and for sending in those questions. We'll get started with a question that has come in. What are the advantages of CF3, Nivesh, over DLS and laser diffraction?
Thank you, Brian. So DLS and laser diffraction, or LD, they offer a size distribution rather than separating the particles and then analyzing them. In such cases, basically the mean size which they offer that may not be as accurate as a representation of the entire population in the sample. CF3, on the other hand, it is capable of separating the particles first based on their mass and then measuring each peak separately. And that gives a much more accurate result for each population in the sample.
Excellent. So one of the questions that has come in from the audience is asking questions about lipid nanoparticles or liposomes. And they're interested to know if Shimadzu has any existing applications in that space.
Yes, absolutely. We do have applications for that, and we have them on our website also. You can access them over there, and we have a few applications already at the bottom of your screen where you can download a few materials for these applications as well.
Excellent, thank you. Another question that has come in is, what are the benefits of the different detectors that can be used with CF3?
Yeah, that's a great question as well. So think of CF3 as a separation technology or a unit, just like a column. You can select the detector according to the application, just like in an HPLC. For example, if you want to know about the particle size or the molecular weight of your sample, MALS or multi-angle light scattering is the best option. Refractive index detector and the fluorescence detector can also be used similarly. Specifically for the fluorescence detector, you can set the excitation and the emission at the same wavelength and use it as a light scattering detector, basically. So several detectors can be used with CF3 in this way.
Excellent, excellent. Another question has come up about the concept of fractionation, which was mentioned as potentially being after the separation by CF3. And the question is regarding the highest concentration factor that can be achieved during that fractionation.
So fractionating samples with CF3 is possible. They would typically depend on the initial concentration of your sample and what is the concentration of individual sizes in your sample population. They would not be very diluted, but they can be further processed to increase the concentration by evaporating the solvent in which they are fractionated.
Yeah, I would assume that because we do have to transport the separated particles off of the CF3, that ultimately, like with LC, this is going to be a dilutive approach that's designed to get them into a pure fraction. And as you indicated, Nivesh, then it would be a matter of dry down to increase that effective concentration. All right, I think we have time for a couple more questions here. One of the questions that has come up a couple of times is, other than in pharma and medicine, are there other applications that centrifugal field flow application would be suitable for?
Yes, absolutely. So CF3 can be used for the analysis of any nanoparticle sample for the size or molecular weight of any polymers and such. Nanoparticles are also used very heavily in the semiconductor manufacturing industry. And the ability to determine molecular weights has application in the chemical industry for polymers and similar applications as well.
There are a couple of questions coming in as well about the idea of agglomeration and detecting potentially aggregates versus individual nanoparticles. Can you speak to that, Nivesh?
Absolutely, that's a great question. So like with any other particle measuring technology like DLS or laser diffraction, your separation here is based upon the property of the whole. If it's an aggregate, it'll be considered as a single particle and it will be separated accordingly. But the difference from the other technologies, the advantage of CF3 here being that you can fractionate that sample and you can view it under a microscope, under a scanning electron microscope or a transmission electron microscope, and see whether those particles are in fact single particles or whether they're aggregates of such. So you will know the morphology of your actual sample, whether you have separated aggregates or single particles.
Excellent. There's also some questions about controlling software. Can you speak to the software that's used to control the Shimadzu CF3 system?
Absolutely. So this is controlled by our one software that we offer for all our instruments, lab solutions. And this is a simple upgrade in the lab solution software that we use for controlling the CF3. And the same platform can also control the front end, the LC, that is feeding into the CF3.
Excellent. One of the other questions that has come up is, are there any requirements or limitations for the solvent systems that can be employed using the CF3?
There are no such limitations, but typically the applications that are used for nanoparticles, they are typically employing aqueous solvents, but there is also some use of organic solvents that we can use with this. There is no such limitation for the solvents as such.
Excellent, excellent. Well, certainly I do see that we have a few more questions, but unfortunately we are coming quickly to the end of our allocated time for this webinar. So I do want to thank everyone for all of their wonderful questions. Because we don't have time to answer each of these questions within this forum, we do have your contact information if you've submitted a question through the widget, which you can continue to do even after we are done with the live portion. And we will reach out to you individually for each of those questions that we were not able to answer in the live session today. But once again, I do want to thank Nivesh Mittal for his presentation and for the audience for attending and participating. We will send everyone who has registered an email link to view the recorded version of the presentation anytime afterwards, which will also include this Q&A session. So we encourage you and your colleagues to attend our upcoming webinars and take a look for that registration link through the e-newsletter. And again, thank you all for participating in today's webinar about the Shimadzu CF3, and have a great day.